not getting this? You know, it's a right angle. It looks like a flat star on each end. Cooper.
Good morning.
Morning.
Oh, just dropping my son off at the bus.
Oh, fun for you.
Not really, but I'm back. It's cold out there.
How old is your son?
Oh, he's 15.
Okay.
Yeah, and normally I'd make him walk, but it's cold. So
Is it really cold?
to-
I know you're more.
I don't
I
know.
think you guys are more up north than I am, and so I think I've
Oh,
always.
I'm down near the Loudoun County border near West Harper's Ferry, but I'm also up
I
kind of in a
kind of
mountain.
know. I kind of know where that is. Maybe.
Yeah, I'm west of Frederick.
Oh, I know. Yeah, I definitely know where that is because I thought, no, I've seen Harper's Ferry.
It's beautiful.
Is that part of Pennsylvania or are you in Maryland?
So I'm where Maryland, Virginia, and West Virginia meet. It's like a little dot.
Okay,
That's where.
because, yeah, I've been there. I've been there a couple times actually.
Yeah. So I chose to go to Hack because they have the program that I want to apply to, the medical sonography program.
Okay.
And but anyway, I was going to Nova in Sterling, Virginia, which was like 45 minutes away. But Gettysburg is also 45 minutes away from me. So I was going to the Gettysburg campus before COVID.
Okay.
You know.
All right.
Anyway, that's story. It's boring.
Yeah, almost. I mean, I can say that, you know, I've been to a very small percentage of places here in this side of the US, but I've been to Harper's Ferry probably about three times.
Yeah, it's so pretty.
It is. I don't do it now, but I used to take care of a friend's little boy, and she lived in, was it Morgantown?
West Virginia.
Yeah. And so, and is that close to, that's fairly close to Harper's Ferry, right? No?
I don't know, Martin.
It might not be Morgantown. I'm trying to think where it was.
Is.
It's not Morgantown.
Shepherdstown's really.
I think it's Shepherdstown.
Yeah, Shepherdstown's very close, and it's
Right.
really. And Frostburg University's there.
Right. Okay. Yep, that's it. Because I used to, so I'd pick him up and we would go to Harper's Ferry for a little bit just to spend time and wander around.
Yes.
And
So
it is really, really pretty.
Yes.
And
I live like.
Where do you live? Like where?
Oh, the Appalachian Trail runs through my town here.
Oh, that's awesome.
Yeah. I live in the only part of Maryland that Appalachia runs through. So there's like this really tiny part of Maryland. The Appalachian Mountains runs through, and I'm in it.
What is it called again?
I live in a town called Knoxville, Maryland, but most people know Brunswick, Knoxville.
Okay.
Yeah, it's quiet. I'm from Northern Virginia. I'm from the big city, but I moved up here to be near my mom. She's my neighbor. So
that's cool. I may have to come out and visit that area because now that I know it's there, and I really enjoy walking around Harper's Ferry.
Yeah, it's gorgeous. In the summer, we have like kind of like a tourist season where we have river rafting and kayaking and zip lining and whitewater rafting. Although there's some crazy people, I swear, that have been out in wetsuits on the river, and I'm like, that's insane to me. It's too cold. That's too fanatical.
I went kayaking maybe twice in my life, and I want to get a kayak because that's just fun to do that kind of stuff. The first time I almost died, but it was still fun up until, I mean, I didn't even realize how close it came to death until after I was told about it. But I ended up on an inflatable kayak and I was with a guy that I don't know, I don't even remember his name now. And we went over a waterfall on accident, and he thought he would do if he did the manly thing and jumped out, I would stay in the kayak. And that didn't happen. When he jumped out, the kayak flipped over and I ended up in the water, and I ended up underneath the kayak. And it was amazing how rational and how logical your mind is in that moment, because it happened so fast. And the next thing I knew, I was in the water, underwater. And I remember, I remember putting my hands above my head and thinking, this is the bottom of the kayak. And I thought, you've got to get so that you're above the kayak. You have to shift the kayak a little bit and get your head above water here. And so I remember trying to like shift it. And then I remember, coming up on top, and I'm wearing a life jacket. And so I thought, okay, just float until somebody grabs you out, gets you out, because what else are you going to do? And I guess when the thing flipped over, I came within 6 inches of hitting my head on a really big rock. And our guide, I mean, I never even saw it because it happened so fast. The tour guide that we were with just paled when he watched it. And I'm just amazed at how rational my mind became. I wasn't worried in the slightest. I think I probably was a little worried when I felt the kayak above me, but I also, didn't, just kind of, I knew what to do. This is what I had to do to get so that I wasn't underneath the kayak.
Yep, survival skills.
Yeah, I want to do it again. Like, I've done it since then, and I got blistered all 10 fingers of my hands and worked muscles I never realized I had. But I had such a good time.
So fun to get out there. You might enjoy river rafting where you just float. It's a lot easier and more relaxing.
Really? I'll try. I'll think about that next time. I'm doing something a little different, and I want to tell you what this is. So if you had me for 122 last semester, we had chapter practices, and I felt like my 121 students last semester I gave them too much work. I gave you too much work. If you think I give you a lot of work now, I gave you a lot more last semester. Especially if you had me for lab and lecture. But when what I took out were these chapter practices. And what they are full of questions. So I give you almost all the questions that the chapter that McGraw-Hill makes available to me. And I tell it to randomly select a pool of those questions, randomly select a set of those questions and give them to you. And that's what I took out this semester. And I had a student in this class ask me if it was possible for her to get those again. And I had to think about it a little bit, because how is it going to mess up the point scheme? And if I did give them to her, I can't make it required for everybody. How am I going to do this? And what I've decided to do, and her explanation, she's here, but she's going to remain anonymous, obviously. She asked me if I would do it, because she learns best by constantly doing problems and learning what she knows, being able to determine what she knows and what she doesn't know, and then getting feedback on them. And so I set it up. And because if I set it up for her, the only way that I could really do this and do it in a fairly convenient manner was to set it up for everyone. And so if you go into McGraw-Hill Connect, actually, you don't even have to go into McGraw-Hill Connect. If you go into our class site, if you go into our class site right down here, it says extra credit chapter 18 practice. And these will be open. I've set up the first 3 chapters. It does take me a little, probably about 30 minutes to a half hour to set each one up. So bear with me. If you really like them, be patient with me as I get them all set up, because it's normally this is something I would have set up at the beginning of the semester before school started and not afterwards. So I only have 18, 21, and 19. But if you click on here, extra credit chapter 18 practice questions, click launch. And what it is, I'm gonna see if I can go into student view. Can I go into student view preview? Is this going to give you a random pool of the 174 questions? It's going to give you 64. And it asks you, and of course with me, it gives me the answers, but it asks you just like you would on the exam or the quiz, but it's a lot more questions. And they're not worth as much because first off, it's extra credit. And you can do this as many times as you want. You can complete this and do it as many times as you want. And do it over and over again. Bear with me here. Do it over and over again up until May, when the semester ends. And they are worth, because it's extra credit, they're worth 5 points, which I figured, I think for your class, the total for the semester is 1,400 points. And so I decided that an additional 60 points was not, that was extra credit that I could do. So that's what this is. I won't round up the points on it. But they will add into your final grade, and you can do them or not do them. But for this particular student, because of the way she learns best, she really likes this. And she knows who she is because we talked about it the other night. but she did actually exceptionally well in my 121 class last semester. And so if she's telling me that this worked really well for her and she did really well last semester, it's worth it to put it back on. Claire, what was your question or your comment?
You answered it. I was gonna say, is it not, it's not required, right?
It's not required.
Okay.
But you have them if you want them. And you can do them over and over again up until the end of the semester. Are there questions? Other questions, comments? Okay. You guys, I would love to see some cameras turned on, just so you know, so I don't feel like I'm talking to myself. But you don't have to. You're not required. I just feel like I'm talking to myself when I'm looking at a bunch of names. Let's get into this. So we will finish the lymphatics today. In lab, if you remember from last week, I decided not that first off, we're just getting further and further ahead in lab. And we're not behind in lecture, we're just getting more and more ahead in lab. And rather than having to introduce new material or really new material all the time that we may not cover for another week and a half to 2 weeks, I decided to do something differently. And you can look through these after class if you like, before we do them, but I put them under the Wiley lab exercises. Why did I do that? Because we were going to do them in lab. I will be assigning case studies later in the semester that I'm going to ask for you to do on your own. But right now, these will do in lab, and it'll give you an idea of what's expected with regards to your answers for lab. If you want to, feel free to read over them before. And some of the answers you may not know. Some of them you will know. And a lot of times what case studies are doing is they're bringing everything that they're going to span multiple chapters sometimes and ask you to apply what you're learning to real case scenarios. But let's jump into lab so that we have a fairly good idea as to what we're looking at in this CAR-T immunotherapy case study. If we finish this, I didn't realize just how far along we were. If we finish this, I will upload what I have of the heart. That isn't a lot. So we may finish this up earlier than I planned on.
I have a question.
Go ahead.
Were you like changing some of the dates in like some of the Connect and Wiley assignments last night?
I was. Was it in this class? I don't remember changing them in this class, but could I have done it? Maybe. What did you see that was changed?
It was like either like some of the heart Connect assignments or the lymphatic systems Connect assignments. Like I did
extending out the deadlines?
Yeah, you like sent it to like February 17th.
I did. The reason I did that is because I a lot of students, because when they see that deadline, they'll take a quiz, chapter quiz, even though we haven't finished doing it in a lecture, and get a poorer score than if we'd finished in a lecture. And I don't want to put you in that position. So I try to set them up so that they are due a week after we finish them in class. And so I set some of them up so they would do that now. And that's why I did that. And I may still push them back. I didn't think about the fact that I'd done it in here too. I knew that I did it in my 121, but I didn't think about the fact that I'd done it in this class. I'll tell you, I get really kind of worn out by the end of the week. And I was at Stevenson University trying to get some stuff done for my classes there. And it ended up not working, which just made me frustrated as all get out. But because I was waiting for that to finish, what I was working on, I started, because I don't like to just sit there, I started working with the deadlines in here, and that's probably what you saw me doing. I was just pushing them back. I didn't push anything forward. Kayla, do you have any questions or problems with it?
No, I was just curious. I have no problems. I was just curious. I'm like, oh, okay.
Yeah, I don't want you feeling like you have to do a quiz like a week before we've even finished it in lecture. And I know there's students who do that and they won't ever contact me about it. They'll just do it. And they'll get a lower grade. And I don't want that to happen. The quiz is not that important that I want you to get a lower grade just to meet a deadline.
I have a question.
Go ahead, Martin.
Wait, so are we done the lymphatic system?
No, we're not. We'll finish it today.
Oh, okay. Because I took the quiz, that's why.
You can retake it too. Did you take both of your attempts, Martin?
No, I just took one.
You can redo it again.
Okay.
I think that was the reason, because wasn't it due last night? It was, wasn't it?
Yeah.
Okay, that's the reason I extended it out, is because we weren't done with it. And that's the reason I extended it out. Does that make sense? Now I understand why I did what I did.
Yeah, that makes more sense.
Okay, if you used up both of your attempts trying to take it last night and did not, and would like to do better, email me or text me and ask for a third attempt and I'll give you another attempt. Okay. Yeah, I'm trying to remember just why I did what I did last night. And now I remember, and I did it for several assignments because I didn't want that to happen again, or not happen as easily as it's been happening. All right, let's come back to about right here. Are you gonna come say hi to my students, Ruby? You guys want to see my really big mammoth of a dog that we've been watching? All right, so I'm not gonna be able to see her very well. I can't see her very well. Ruby, say hi. Ruby, look up. Yep, there she is. Okay. It's just been she and I all week, and she was our neighbor's dog, and all of a sudden she's sharing my room with me, because she was not in my room initially when we brought her downstairs from upstairs. But then every time I would start to shut my door up at night, she would start looking at me like, wait, are you going to bed? And so I was like, okay, you win. And now she's sleeping on the floor in my room because I can't do that to her. And she knows it. And she's really a good dog. She'll sit there and bark in your face until you wake up to take her outside. It's a very good wake up call. All right, let's get going. Yeah, I think we'll finish this. We'll see. I didn't realize that we had this much left, but let's go. So actually, let me back up just for a minute. So if you look, I think I inserted these afterwards, and that's maybe a good reason that I'm skipping back just a little bit. I put these charts in here last week. And what they are these are all agents involved in inflammation and non-specific immunity. What I would do if I were you is I would make flashcards of these. Some of these are not if you look at the stars here, these are things that are also involved in specific immunity. And so I would make flashcards of these. You've seen some of these, fibrinogen. Remember, that's involved in the blood clotting process. Well, it also has a role in inflammation. The complement system. Colony stimulating factors. We'll talk specifically about what they are, but what they do, think about what it would be to be a colony stimulating factor. If you're stimulating a colony of bacteria or a culture of bacteria, these stimulate their numbers and increase their production. Some of this we will talk more about today also. But it's just a summary of the functions of all of the major agents involved in inflammation. Well, let's see. So two main classes of adaptive immunity. Antibody-mediated or humoral antibody-mediated immunity and cell-mediated immunity. Antibody-mediated meaning that B cells secrete antibodies that float free in the blood and can go out and perform their immunic reactions without direct contact with the pathogen cell. And we'll talk specifically about how that happens in a moment here. But so it's antibody-mediated immunity. Cell-mediated, because in cell-mediated immunity, direct cell contact is necessary every time for destruction of the pathogen. And B lymphocyte immune or antibody-mediated lymphocyte immunity always involves antibodies and plasma cells and the B lymphocytes. Cell-mediated immunity always involves activation of T lymphocytes. And antibody always requires antibodies, obviously. And cell-mediated immunity directly attacks unwanted cells. Questions so far?
So,
If you go ahead, Martin.
for the T helper cells, like on the diagram,
We will spend a lot of time talking about them today. Okay. Because they are actually a major crux of the immune system. And so they're involved, we'll talk about how they're involved in we'll We'll talk about how they're involved in the innate inflammation system, and they're also involved in activation of B lymphocytes and other T lymphocytes. Does that answer your question a little bit?
Yeah.
Okay. So just looking at this really simple diagram, and if you look here, do you see the TH? See the TH cell right here? That H there, and I know your screen is probably a lot smaller. Let me zoom in just a little bit more. It gives you an idea. See the TH on there? Somebody say yes.
Yeah.
Okay.
Yeah.
What that means is the TH, the H stands for help, helper. So this TH cell works on B lymphocytes or B cells, and it will also work on cytotoxic T cells. And it also works to stimulate the production of more memory T helper cells. It's a major crux of the immune system. And it's also why the AIDS epidemic is so nasty. And why the AIDS virus is so nasty is because it infects T helper cells. And that is specifically attacking the immune system by going after the T helper cells. So antigens are engulfed and displayed by phagocytes. We talked about that in the last chapter. We'll talk more about it. Excuse me, talked about it last week. We'll talk more about it today. But antigens are engulfed and displayed by phagocytes. They activate T cells and B cells. The intact antigens or free floating antigens can activate B cells. Antigens that are displayed by infected cells activate T cells also. So be patient as we go through some more detail about both of these processes and all of these processes. So humoral or antibody-mediated immunity protects the body against extracellular pathogens, so things that are outside of cells, and their toxins. Cell-mediated immunity protects the body against intracellular pathogens. And antibody immune or humoral immunity requires the secretion of antibodies that or allows for the secretion of antibodies that can float in the blood all by themselves and attack any antigen out free in the blood. Cell-mediated immunity has to have cell-to-cell contact, but it also is capable of killing intracellular pathogens like viruses and cancer cells. So this adaptive specific immunity, it's mediated by B and T cells, and it targets a particular pathogen. It's specific. It's not like the innate immune system or our hitmen, the natural killer cells that just are able to kill everything and anything that looks weird. These are specifically attacking certain things based on what's present on the cell. So, but because it's specific, it's not always ready. It's got to be primed. But once it's activated, it's systemic. And a stronger second response of the adaptive immunity is going to occur. Your first response is going to be, it's going to be slow, but it's going to happen and it's going to be specific. Your second response will be much quicker and faster because you have memory immunity. So the innate immunity, as we talked about last week, the innate immunity essentially holds things off and limits the spread of infection until this adaptive immunity is ready and can step in and eliminate the infection. Questions? So specific adaptive immunity. B cells come from lymphocytes. And T cells come from, excuse me, B cells originate and mature in the bone marrow. T cells originate in the bone marrow, but they mature and differentiate in the thymus. They gain immunocompetence in the B cells gain immunocompetence in the lymphocytes. The T cells gain immunocompetence in the thymus. What that means is when they're immunocompetent, and bear with me here, because I'm not sure we're going to see this word again until the very end. Immunocompetent means that that cell is not immature. It's able to facilitate an inflammatory response and an immune response. So B cells gain immunocompetence in the bone marrow. T cells gain immunocompetence in the thymus. So,
Can I ask
Go
a
ahead.
question?
of course,
Okay, I'm sorry. I just wanted to make sure I got this right.
don't be sorry. I would prefer you guys interrupt me, in all honesty, and it makes me feel less like I'm talking to myself. I see some faces here and I appreciate it. Keeps me from feeling like I'm talking to myself. Go ahead, Zena.
Okay.
What was your question?
So you say that the B and T, so they both originate in the bone marrow.
Uh-huh.
But the T cells mature in the
Right. The T
in the
cells
thymus, yeah.
in the thymus, right.
And then the B cells in the bone marrow still.
Right.
Okay, I just want.
So the B cells originate and mature in the bone marrow.
Okay.
The T cells are going to originate in the bone marrow, but then they're going to travel to the thymus.
Got you. Okay.
And mature there. Yeah, that's a good question. After early childhood, new B and T cells are mainly produced primarily by B and T cell colonies established in peripheral lymphoid tissues like your lymph nodes and your your spleen and tonsils, adenoids. Remember, the only two primary lymphoid tissues are the bone marrow and the thymus. All other organs that are involved in the lymphoid system or the immune system are secondary or peripheral tissues. So B cells then, once they're activated and gain immunocompetence and they go on to facilitate antibody-mediated immune responses. T cells go on to facilitate cell-mediated immune responses. So kind of adding to Zena's question here. B cells and T cells. B cells originate and mature in the bone marrow. T cells originate in the bone marrow, but then become immunocompetent in the thymus. All of these others are secondary cells, are secondary tissues, where these cells can be activated and possibly, and we'll talk about this, stimulate more, they can undergo cloning and increase in their numbers in these other peripheral cells. Excuse me, peripheral tissues. Is this clear? So T cells achieve immunocompetence in the what?
D.
Has their microphone.
Say it one more time.
Somebody has their microphone on. I can hear.
I know, and I think they got a kid who's a little angry at that moment. Tell me one more time, what's the answer?
Said D.
D is in Delta, right?
Yeah.
So one thing that's really important here, if you remember, we talked about blood typing, and the blood typing involves antigens. Well, if you remember, blood typing we use antibodies that are specific for that antigen. Therefore, we need to spend some time specifically talking about antigens here. Antigens are large, foreign, non-self-unique molecules. You know, it's only considered an antigen when it didn't originate from your body. So the antigens on your red blood cells, your body doesn't deem as antigens because they're part of who you are. And you've always had them. They're not non-self. When the immune system stimulates an attack and goes on the attack, it's going after antigens that are non-self, that have not always been with you since the day you arrived on the earth. So antigens can induce an immune response against themselves. In general, the more complex a molecule is, the greater its antigenicity. And I learned something the other night. I was reading a physiology book to give me a little bit of a review on some of this material. The antigen itself is short for antibody generating. That's where the name comes from. And if that helps you to remember it, let me bear with me here just for a second. I may have learned that at some point, but it only hit me again last night that it would probably help my students to remember it. But an antigen means antibody. So it's short for antibody generating. An incomplete antigen or a hapten, think of it as being a half an antigen, hapten, does not trigger an immune response. However, once it's maybe attached to a carrier molecule, it's then capable of triggering an immune response. The antigenic determinants on an antigen are called epitopes. This is this part of the antigen that is able to bind to a receptor on a B cell or a T cell. On the B cells, those receptors are specialized antibodies that are on the surface of the cell. On the receptors of T cells, they have that same binding structure that allows them to bind to epitopes. But T cells aren't going to secrete antibodies later. So the epitopes are the part of the antigen that's specifically recognized and bound to antibodies on B cells or receptors on T cells. This little receptor is called a paratope. So antibodies and epitopes or antigens. This little part of the antigen is the epitope that is capable of binding the paratope or the antibody binding region of the antibody. Antibodies are a construction of two heavier chains and two lighter chains. The heavier chains are the Y shape of the the Y stem of the antibody. The lighter chains are part of the two branches of the Y of the antibody. The lighter chains and the heavier chains are held together by disulfide bonds. The variable regions of the antibodies are the spots that bind to the antigens. We call this the Fab, which means antigen binding. The constant region here is what is going to dictate what happens once that antibody binds to an antigen. And it's constant. We have 5 different classes of antibodies. This region is constant for every class. It's the same structure for every class of antibody. You can see there's a complement binding system. If you remember, one of the ways to activate the complement system is via the antibody antigen structures. Questions about this? Self-antigens. So these aren't antigens in the definition of the word because they're part of who you are. Antigens that can stimulate an immune response are non-self antigens. So self-antigens are the MHC molecules. MHC stands for major histocompatibility complex. Why do you think they're called histocompatibility complexes? What does histo mean?
Belong to histamine
Or what else could it have to do with?
tissue.
Tissue. These were discovered because of transplant rejections. The major issue that comes with transplant rejections is rejection because it's non-self tissue, because they're attacking these antigens. The reason we don't have this problem with blood is because blood is the only cell that doesn't contain MHC molecules. These are carriers that hold the antigen on the surface of the cell. If you remember when we talked about phagocytosis last week, and we talked about how it takes the substance into the cell, and then it chews it all up, and then it takes certain parts of it and puts it on the outside of the cell. This is what carries them and holds them on the outside of the cell, those pieces.
Sorry.
So she was ready to ask a question, and then her dog just started to start barking. What was I going to say? So when you just remember with the phagocytosis, and I told you about that book that I really liked. I still really like that book. I was looking at it the other day. Think of this cell as it eats a bunch of stuff. It takes in this big old parasite or pathogen, it picks it up, it takes it in, engulfs it, digests it down, and then it takes little bits of everything that it just broke down, and like the guts of the pathogen, and then puts it all over its body so that everyone can see what it just ate. And the carriers and the carrier proteins for the way that it presents all this stuff all over its body is these MHC molecules. So they just kind of hold it out for display on the surface of their cell. You have all of your cells do this, even cells that don't take up antigens. They're constantly displaying pieces of themselves on the outside of their cell. We'll talk about why that's important in a moment here. But think of these as just carriers that are holding antigens on the outside of the cell. So major histocompatibility complexes. There's two different types. There are class 1 MHC molecules that are found on all antigen presenting cells. Actually, I messed that up. I knew that the second it came out of my mouth. MHC1 class molecules are found on all cells. All cells. All nucleated cells, they are found there. MHC2 molecules are found on all antigen presenting cells. So all cells that are capable of phagocytosis and then display those guts all over the outside of their cell.
I have a question.
Go.
So for both of these MHC cells, are they both used in humoral and
Yes.
cellular?
Yes. Okay. Yes, and we'll talk about why in a second here. Actually, wait, let me think about it. I'm going to say no. Let me think about this because I think there's exceptions to this. But generally it's through the T cells that they're interacting. It's not through the B cells. Bear with me and let me address that as we go. Is that all right?
Yeah.
Okay. So class 1 MHC molecules found on all cells that contain a nucleus. All nucleated, found, let's change this word here, found on all body cells, found on all body cells that contain a nucleus. So they're not found on red blood cells. MHC2 molecules are found on all antigen presenting cells, so all cells that are capable of phagocytosis. MHC1 molecules activate cytotoxic T cells, which are similar in ways to natural killer cells, except these are specific. Natural killer cells are just non-specific hitmen. Cytotoxic T cells are specific. Helper T cells are activated by the MHC2 class molecules and the antigens present on them. If you look here, here's the MHC molecule is a green here. Is a green here. The antigen is that little orange part. So it's just a carrier antigen MHC molecule that's found on the surface of the cell. Different types of immunity. We have naturally acquired specific immunity, and we have artificially acquired specific immunity. We have passive naturally acquired immunity, and we have passive artificially acquired immunity. Think of active as self-generating. You generated it. Passive means you borrowed it from something else, or you got it from something else. Differences here. Active immunity requires the active immunity means that you're exposed to antigens. Whether they're injected into you or you come in contact with them at some point and get exposed. The person generates them, the antibodies. So you get exposed to the antigens, your body generates antibodies. Your T cells get activated by those same antigens. In immunizations, you get injected with either killed or attenuated pathogens. The only difference between naturally acquired immunity and artificially acquired immunity is naturally is natural exposure. Artificially means giving you a small amount of a killed or attenuated pathogen or their toxins, and intentionally exposing you to the antigen, a small amount of hopefully not too nasty of a amount. This active immunity takes 5 to 14 days to develop the resistance. With active immunity, the duration is long, perhaps to years or the rest of your lifetime. When for Passive immunity, you receive antibodies. You either receive them through breast milk colostrum or because they pass through the placenta. In artificial acquired, you get injected with antibodies. If you're not going to have time, let's say you get bit by a snake and you're not going to have time, it would be great if your body could just develop an immunity against it. But you might be dead before you actually have enough of an active immunity developed against it. Therefore, they take a serum of antibodies from an animal that already generated antibodies against that pathogen, and they give it to you by injection to neutralize those antigens that could possibly kill you before you develop an immunity against them.
I have a question.
Go.
So, which would that be classified as a vaccine?
No, it's not. It's not, because you aren't, this isn't you actually developing antibodies and developing any long-term memory of those. It's not, rather, this is developing antibodies. Active immunity is developing antibodies and developing T cells that have been exposed to that antigen. You don't develop that here.
Okay, so a vaccination would be artificially acquired then.
Yeah, it's artificially acquired. You're right. Naturally acquired is when you're naturally exposed to it.
All right.
You know what? This is a terrible slide for explaining this stuff. I've decided. I don't know how I managed to use it for as long as I have. Give me 1 second here. Because I think I used something different a year ago because I don't like this one. Let's see. I like this slide better. I'm wondering if this is what I've used before, and this is what I need to add to my thing here. Active immunity versus passive immunity. Active immunity is immunity you develop after being exposed to an infection or getting a vaccine. Passive immunity means you acquire it from someone or use something else. Here you're developing antibodies. Here you're getting them from something else. You're borrowing it. Natural active immunity means that those antibodies are made after exposure to an infection. Artificial active immunity means that those antibodies are made after getting a vaccination. You get a small amount of an attenuated pathogen or a weakened pathogen. Passive natural immunity means that the antibodies are transmitted from a mother to a baby via the mother's milk. Artificial passive immunity means that those antibodies are acquired from an immune serum that was developed in another animal. They don't last very long, but they get rid of what's probably going to kill you before you develop enough of an immunity against whatever it is. Does this help? Martin? Was
Yes.
that a yes? Okay, perfect.
I was like,
I will,
I like that slide much better.
you and me both, I don't even know why I have that one because that's terrible. As I thought it was good up until I tried explaining it and it's terrible. If anything, they should be both there, but I'll actually insert this. I'll change the PowerPoint that's on there right now after class tonight and add a slide like this and a figure like this. And I don't think that's the first. I go through a lot of this same material with my physiology students at Stevenson next week. And I didn't realize how bad that figure was. You guys are the guinea pigs because you're seeing it first. Bear with me. That's not what I meant to do. I'm almost sure that that wasn't the figure that I'm using right now was not the figure I used last semester, last year when I taught physiology at Stevenson, because that was terrible. So all of these processes, both passive and active immunity, involve B cells, the humoral immunity, and their antibodies. B lymphocytes are the facilitators of antibody-mediated immunity. Each B lymphocyte has surface receptors for the binding of one of thousands of millions of many different antigens. Each lymphocyte in your body. That you're born with these lymphocytes, they develop, and you have one lymphocyte that's specific for one of millions of antigens that you may or may never get exposed to. Upon binding its antigen, it's kind of like searching for your soulmate your whole entire life, and you may or may never find them. Well, for the B lymphocytes who are lucky enough that they finally meet their antigen that they've been looking for their whole entire life, then they get activated. So on binding with the presented antigen, the B cells will then proliferate. Once they meet their soulmate, they get all excited and they get activated. And what that activation requires is that they undergo cell proliferation. Some of those cells become plasma cells. And if we get a T helper cell that then helps these cells as they get all excited because they finally met their soulmate in the antigen. If we get help from a T helper cell, in addition to a type of plasma cells that are going to secrete antibodies, we'll also be able to generate clones of memory cells that have the receptor for that same antigen. So it's cloning. It's what we call clonal selection. Most are going to secrete into plasma active cells or differentiate into plasma cells that secrete antibodies. If we're lucky enough to get help from a T helper cell, some of those proliferated cells will become clones of memory T cells. So you bind to the antigen, some of the cells become plasma, you undergo this massive state of proliferation. Some of the cells become plasma cells, some of them become memory cells. But now all of these cells that have been cloned from this target being able to bind to this specific pathogen have now proliferated. And now they're all specific for that antigen. So all of these cells are capable of binding that antigen now. This cell, all these cells running around in your bloodstream, one of them is lucky enough to meet its antigen and it undergoes cell proliferation. And some of them become plasma cells, some of them become memory cells. The plasma cells go on to secrete more secrete antibodies that are specific for the antigen that activated them. Does this make sense? Is it clear? If these plasma cells, they don't last for forever. They can secrete a lot of antibodies. They secrete up to 2,000 a second, each of these plasma cells. But the antibodies don't last for forever. The memory cells are now one of these that's been floating around for the rest of your life. And they float around for a very, very long period, if not for the several years and possibly the rest of your life. And when they get exposed to the same antigen again, they're going to undergo the same process and be one of these that then become, some of them become memory cells and some of them become actively secreting plasma cells that are actively secreting antibodies. Allowing for a faster response because you have more of them now. This one cell that you had that you were born with, you now have 4 of them. And chances are you have more than 4. You probably have 100 of them or 200 of them. And so when they get re-exposed to that antigen, all of them start, each of those, some of them start to proliferate, some of them become plasma cells, some of them become more memory cells. This clonal selection and memory cells is the basis behind how vaccinations and boosters work. You're first exposed to they give you a slight variation. They give you a small amount of the COVID mRNA, and I promise I won't get political on you again. But they give you a small amount of that mRNA. That mRNA starts making proteins that it infects some, let's see, how do we say this? It enters some of your antigen, your phagocytic cells. Let me say it differently. Let me not use the COVID vaccine because I'm not sure I really understand how that's going to work. Let me say it differently. You get, unless you have an egg allergy, they give you a small amount of, when you get your flu shot, they give you a small amount of a denatured flu virus. It gets taken up by an antigen presenting cell. Or it actually, let's not say it gets taken up by an antigen presenting cell, because it doesn't. It's floating around in your bloodstream. So in your millions of naive B cells that have never found their soulmate yet, and that's why they're naive, is they've never been activated by antigen. You have an antigen B cell that's capable of binding to that flu protein. It's got the receptor for that flu protein. When you get exposed to it, it goes through this process where some of the cells become plasma cells, some of them become clonate cells. The plasma cells specifically secrete this antibody IgM. We'll talk about these different classes in a moment. And some of them become memory cells. The ones that when you got injected with a small amount of the flu protein, these plasma cells start secreting IgM antibodies, and they eliminate the flu protein altogether. It's gone. Ruby, stop it. And in addition to stimulating a production of plasma cells, you now have a colony of memory cells that are capable of binding to that antigen later, if you see it again. Now, let's say you have to get a booster of a flu shot, just like you get a booster to the COVID. It works the same way. You get a booster of the COVID vaccination. Because you have these memory cells floating around, and now rather than just having one naive antigen B cell that can bind to that, you have more than one.
Does she have to go out?
You know what? I took her out right before class, and that's what I'm wondering. Why is she barking out there?
Take a break. No.
I think, you know what, Claire, if you guys don't mind, let me see what in the heck is wrong with her. And I'll take her out one time, and if she does it again, I'll shout at her and lock her into her room. But you guys, I apologize. If it was, my mom is gone until this afternoon. And so it's just been her and me, and normally she doesn't do this. I've been lucky that she hasn't done this in my classes yet until now.
Yeah, well, maybe she has to go potty, and I need a potty break too.
Oh, no, hey. Let's do it right now. I'll see you guys in a minute.
Okay.
Ruby, you stinker butt. You know you're a pill. You need to go outside. You know you just kind of have a little animal. You're a turkey. You need to go outside. Let's go. Come on. want a dog treat? Come on. Come on. You want a dog treat? Come on, Ruby. Hey, come on. Okay. All right, I apologize. And yes, she did need to go out. It's great that she's so dog trained, but it's been a long time since I had a dog this big, and she can wake up the whole house. Any questions about how this works? So with your first injection of a vaccine, the special little antigen binding naive B cell has been floating around your whole entire lifetime waiting for its antigen to show up. Now binds to that antigen, and some of it proliferates into plasma cells. And if there's a T helper cell around that can expose it to a protein called interleukin-2, some of the cells that proliferate from that B cell will become memory B cells that are selected for that particular antigen also. The next time you're exposed to that antigen, you get IgG antibodies. And we'll talk about specifically how one works versus the other. But IgM are first, the primary immune response. IgG are the second. And because you have a lot more, once you've been exposed the first time, you have a lot more memory B cells around. They're going to secrete a lot more antibody than you had initially with your first response. So different classes of antibodies. You have IgA, which actually is a dimer in its structure. It's found as a monomer in the blood plasma and mainly as a dimer in the mucus, tears, milk, saliva. So this is what we find in our mucus, those slimy layers that like to trap up pathogens that are trying to enter via our digestive system or our respiratory system. They sometimes also form trimers and tetramers, but they prevent pathogens from adhering to the epithelia because it catches them up in all that slime. You imagine, think of mucus as being stuck in a swimming pool of slime. IgD is actually the receptor found on B cells. No, you can't. I'm not taking you out again. Memory, so it's a receptor that's found on intact B cells, and it functions to activate the B cells. So it's never found actively secreting, it's just found on the B cells. IgE is the protein is an intact protein found on basophils and basophils. If you remember basophils, they're the leukocytes that are going to secrete heparin and histamine. When they secrete heparin and histamine, The only way that they secrete that is by being triggered to do that. The receptor that they have that binds to an antigen is IgE. These are important in your immediate hypersensitivity reactions. A major example of immediate hypersensitivity allergies. And in attracting eosinophils to parasitic infection. IgG constitute about 80% of the circulating antibodies. They're the predominant ones secreted in a secondary immune response. IgG and IgM are the only antibodies that are capable of complement fixation. That complement cascade, they're the ones that are capable of activating complement. Remember that that's a classical pathway for activating the complement cascade. IgG are also the ones that are capable of crossing the placenta and confer the temporary immunity on the fetus. These are also the ones that cause, if you don't get the RhoGAM shot and you're Rh negative and you have an Rh positive baby, these are the ones that are involved in hemolytic disease of the newborn. IgM, if you think of it, this is the way that I think about it, so that I remember that this looks like a dimer and this looks like a much bigger one, a pentamer. IgA has 2 branches, so that's the reason it can look like a dimer. The A has a branch here, the A has a branch here. Every letter A has two branches off it. IgM has 3, so it's got the most amount of branches. It's capable of being a pentamer. Constitutes about 10% of the circulating antibodies in the plasma. It can also function as part of an antigen receptor on B cells. The pentamer occurs in the plasma, blood plasma, and lymph, and it's a predominant one secreted in the primary immune response. It's very capable of agglutination because it's got 10 different ways that it can bind antigens. So of course it's going to easily clump them. It's the one that's involved in binding up A and B antigens in blood and causing them to clump.
Question.
Go ahead.
For the IgM and I think like those last two,
IgG.
yeah, what are they used in?
The IgG are first off, they're the ones, remember we talked about the complement cascade last week?
Kind of. I'm like,
Okay. I know I don't have time to go through it again, but it was back here. It's amazing. Leo, stop, buddy. Sorry, having a little bit of a cat dog fight here. The complement system is capable of neutralizing and destroying things fast, but it has to be activated. One of the primary ways, and the classical way that it's activated, is via antigen antibody activation. IgG and IgM are capable of doing that. None of the others are capable of doing that.
so they're both a part of the complement.
Well, they're capable of activating it. None of the
Okay.
others are capable of activating it.
All right.
It's an important thing that when it happens, it destroys things. You know, it's part of the innate immune system. And it is a big deal in that it easily and aggressively destroys things. And IgG and IgM are the only ones who are capable of doing that and activating that cascade.
Okay. And
IgG also is the one that's capable of crossing the placenta and can confer an immunity on the fetus.
right. So does IgM come before IgG or?
I don't know which one came first.
Okay.
I couldn't tell you that. All I know is that it's one of the 5 different types. IgM, remember we talked about how one thing that antibodies do is they make things clump up, especially with blood typing. When they bind to the A and the B antigens and induce them to clump. Well, if you look, each of these Y branches here is capable of binding an antigen. Each of them. And what that means is that each of these pentamers of IgM are capable of binding 10 of the antigen. Not different ones, but 10. They're all the same antigen. And what that means is that you essentially, if you think about it, they look like little crabs. And if you imagine you're a pathogen and you get into a body, you're all excited because you made it through their first line of defense and you're going to go and build up colonies and anything. And the next thing you know, you've got these little stupid little crab things trying to bind to you. And you have to drag them around with you as you try to do what you came in to do. They're binding to you and kind of pulling you down. And then of course they bind to your body, or even the same one binds to your body, and you're stuck with each other because this little crab thing is not letting go. Now pretend that this little crab thing is attached to 5 other of these little crab things, and therefore you have to, rather than just dragging one around, you have to drag around all 5. Let's talk specifically about what they do now. Well, that's one of the ways that they neutralize things. Martin, did that help?
Yeah, sort of.
Okay. So how do antibodies physically neutralize and invade microbes? One way is they cause agglutination and precipitation. The next way is that they can amplify innate immune responses by activating the complement system, enhancing phagocytosis by acting as opsonins. Remember that opsonin meaning making something look more delicious. So if you thought cake was really good, you like eating cake, but you like it even more with icing on it. Opsonins are icing on the cake. They make it look even better than it did before. So your phagocyte that's really, you see something that you think you might want to eat, but it would be better if it maybe had a little salt on it. And the opsonins are that salt that make it look even better than it did before. So that's one of the ways that antibodies can enhance phagocytosis. They also stimulate natural killer cells. When the natural killer cells see that antibody, they kill it. They kill the cell that it's attached to. Remember, the natural killer cells are the hitman of the immune system. So antibodies can neutralize antigens by preventing these chemicals from interacting with susceptible cells, or by binding foreign cells by agglutination. You just start clumping them all together, and these are like little crab things. You can't get rid of them, and they just hang on to you. And they're going to impede your functions just because you have to drag them all around. Um, so agglutination is the clumping of antigenic cells, and precipitation will occur if that clumping complex becomes too large that it can't stay in solution. Antibodies can also neutralize antigens by activating the complement system. Ultimately can cause rupture of the cells. They also neutralize antigens through this opsonization, making it look better than it did before, enticing the phagocyte more than it did before to eat it. Or stimulating our natural killer cells to lyse the cell. Now, if you remember, we talked about this before, how natural killer cells, these are our hitmen. They go up, they look at a cell for a minute, and sometimes they sit there and look at the cell. If they're initiated by an antibody, the antibody itself being bound to something stimulates the natural killer cell to release perforants, and then granzymes enter those perforants, those little pores, and the enemy cell dies by apoptosis, and then it's engulfed by a phagocyte or macrophage. One group of proteolytic enzymes secreted by natural killer cells is what?
Is it B?
Say it one more time.
Is it B?
B is in boy?
Yeah.
No. You're close. So cytokines they're signaling proteins. Let me give you a clue. Anytime you see the word an enzyme, they almost always end in ZYME. So what?
C.
It's C. So if you look back on this pathway, so the perforants, they insert these perforants. Here's the natural killer cell. It goes up, it recognizes that there's something wrong with that cell. Well, it doesn't know what it is, but it knows there's something wrong with it. And so it releases these perforants, which make up these little pores on the enemy cell, and inserts into the surface of the enemy cell. And then granzymes are released that go in and degrade the enemy cell enzymes, and this enemy cell dies. B cell receptors and T cell receptors are not that different in their receptor. Their structure is a little different down here, but they both have antigen binding sites, except the T cell receptor only has one. The B cell receptors have two. But in structure of the antigen binding site, they're the same. So yeah, I already talked about that. T cell lymphocytes carry out cell mediated immunity. What this means is that they directly have to come in contact with the cell to be activated. They have to come in contact with an intact cell. There are several types of T cells. There's cytotoxic T cells, which are similar to natural killer T cells, but they're specific in their functions. There's helper T cells, which help mitigate and enhance the immune responses. And then there's regulatory T cells, which are the ones that we know the least about, but they're involved in bringing down the inflammation when the infection is over. T cells don't secrete antibodies. They directly bind to their targets when they kill them. Just like B cells, they're clonal and antigen specific. They're proliferated through the clonal selection process, just like B cells. They have to bind to their antigen before they proliferate. They acquire their receptors and are trained in the thymus. Yeah, I'll talk about that later. I thought of something, but I'll wait on talking about it until later. T cells are activated for a foreign attack only when it's on the surface of the cell that the cell carries foreign or self antigens, and cell contact is required. So not free-floating antigens in this case. They have to be on the surface of a cell. The antigens that they're going to attack and bind to have to be on the cell. They learn to recognize foreign antigens only in combination with a person's own tissue antigens in the thymus. I told you how much I like this book and I highly recommend it. But one of the things that he talks about is if you think that college is hard now, imagine being a T cell in the thymus, because there, if you don't pass the test, you die. And right now, school may be hard, but you're not going to die if you get a wrong answer. In the thymus, if you bind to a self-antigen, you're dead. So this is where the detection between self-antigens and non-self foreign is determined by T cells. And they're not allowed in the bloodstream until they've been tested. And if they fail that test, then they get killed. So a few days are required before T cells are activated to launch a cell-mediated attack. So remember that the innate immune system is holding things off until the adaptive or immune system is ready to go. There's two basic types of T lymphocytes. There's CD8 or cytotoxic killer T cells that destroy host cells that harbor anything foreign. They release chemicals that destroy targeted cells. There's CD4 cells, which are mostly helper T cells that modulate the activities of other immune cells. And they secrete chemicals that amplify the activity of other immune cells. They amplify the activity of B cell growth factors, T cells via interleukin-2 and macrophage migration inhibition factor. These are all chemicals that they secrete that are going to stimulate B and T cells and phagocytes, macrophages. The differences in the MHC molecule is what is going to stimulate. If it's an MHC1, it's going to be stimulated by the cytotoxic T cells. So MHC1 only stimulates cytotoxic T cells. T helper cells are only stimulated by antigen presenting cells that display MHC2 carrier, antigen carrier molecules. Major differences between CD4 and CD8. CD4 are T cells that express a CD4 glycoprotein. So this is a self protein on the cell that indicates that that particular cell is a T helper cell. So they express a CD4 glycoprotein on the cell membrane. Cytotoxic T cells, or CD8 cells, just express the CD8 glycoprotein marker. Just like your A and B antigens, these are those type of antigens. Helper T cells display the CD4. Cytotoxic T cells display CD8. I'm going to say something really quick. Let me back up. To help with this CD stuff, CD stands for cell differentiation. So this is just a marker on the cell that indicates this is what the identity of that cell. CD4 is the glycoprotein that says that cell is a helper T cell. The cell that expresses it is a helper T cell. If it expresses CD8, that cell is a cytotoxic T cell. So CD4 cells, also known as helper T cells. CD8 cells, also known as cytotoxic T cells. CD4 glycoproteins serve as a T cell receptor. CD8 serve as a T cell receptor. CD4 recognize antigens on the surface of antigen presenting cells. CD8 recognize antigens on the surface of all nucleated cells. CD4 cells recognize antigens that are presented with MHC2 molecules. CD8 molecules recognize antigens presented along with MHC1 molecules. CD4 cells induce other immune cells to trigger an immune response by secreting cytokines. Cytokines are signaling molecules. CD8 cells induce cell death of virus-infected cells or tumor cells, either by cell lysis or apoptosis. So how are T cells activated? T cells are pretty dangerous little buggers. And therefore, there's a couple steps with them getting activated. because we don't want them activated unless they really have to be activated. We don't need them randomly activated. Remember, those T helper cells help a lot of different things. So once they're activated, they're going to initiate a much stronger response. Therefore, we want there to be more steps involved in activating them. The first thing that happens is that the T cell receptor binds to an antigen presenting cell that's displaying the antigen that its receptor binds to. Then, just to make sure that it really has bound its antigen and that it's indeed an antigen presenting cell, it binds to a second protein on the antigen presenting cell. Just to verify that this really is a true thing and that this is a sure thing. As soon as this co-stimulation occurs, clonal selection then occurs, where some of the cells become effector cells that are like plasma cells, but these aren't going to secrete antibodies. And some of the cells become memory cells. This is the same for both cytotoxic T cells and helper T cells. Some of them are going to become memory cells. Some of them are going to become effector cells. Next, the effector cytotoxic T cells are going to attack and destroy abnormal cells with a lethal hit. And T helper cells are going to secrete interleukins that stimulate multiple forms of attack. I cannot remember if I have a slide in here about interleukins or not. So I'm going to say something about interleukins. Interleukin.
Real quick, why is there so many of the clonal selections?
Why are there so many clonal selections? Because your body contains receptors, it contains B cells and T cells for any antigen your body is ever exposed to. However, they're not all activated, and you only have one or two in your whole body. But if you get nailed with a major, like a flu infection, you need more than one cell to be on the attack at that point. So what happens is when that B cell or that T cell actually comes in contact with an antigen, it gets stimulated to proliferate. Once it finally meets the one antigen in the world that it will ever bind to, it then undergoes clonal selection, which is important if you're trying to fight off a virus. You need more than one or two cells that are fighting for you, right? Lauren, does that make sense? Or Lexi, does that make sense?
Yeah, that makes sense.
Okay. Interleukins. If you remember, what's the scientific name for leukocyte? I already gave it to you. What's the scientific name for a white blood cell?
Leukocyte.
Leukocyte, right. All right, that was funny. I can't believe I said that. Interleukin, meaning they move, these are signals that interact and allow different leukocytes to interrelate. They work and mediate in between leukocytes. That's what interleukins are. And there's lots of different ones. So major distinctions, because it is this MHC1 versus MHC2, major distinctions between these two complexes. MHC1 molecules are found on the surface of all nucleated cells. They're recognized only by cytotoxic T cells. And the CD8 co-receptor is going to bind the two cells together. When linked in this way, the cytotoxic T cell can then destroy the body cell. No, you are not going to go down there and harass her. I don't think so, buddy. No, you're not allowed to do that. So CD8 co-receptors link the two cells together, and they're linked in this way so that the cytotoxic T cell can destroy the body cells if invaded by foreign or viral antigens. The MHC2 molecules are found on the surface of immune cells with helper T cells. So the MHC2 are what are going to trigger the activation of helper T cells. Do not go over there. Go over there, I'm going to push off my lap again. Don't do it. MHC, so they're found on all antigen presenting cells. And they interact with dendritic cells, macrophages, and B cells, and helper T cells. They're recognized only by helper T cells, and their co-receptor, CD4 co-receptor, is going to link them together. To be activated, the helper T cell must bind with the MHC2 APC. The APC, remember what APC means, antigen presenting cell. So the MHC2 bearing APC, after it's activated, the helper T cell must bind with the MHC2 bearing B cell with the displayed foreign antigen to activate them. Questions?
So since the MHC1 cells are only activated by cytotoxic T cells,
Right.
does that mean that they're only a part of the cellular, or?
Well, yeah, they're only part of cell mediated because, yeah, yep, that's right.
Okay.
And so the major difference is CD8 cells, they're cytotoxic T cells, which means that they kill cells. They kill virus infected cells, they kill tumor cells. They, that's the job of the cytotoxic T cell, is to kill cells. So when it sees that there's like viral particles attached to one of these MHC1 molecules, they know that that's a virus infected cell and they can kill it. Helper T cells can't do that. If it's a cancer cell and it's got a cancerous antigen attached to it, the cytotoxic T cell can kill it. Helper T cells only are working with antigen presenting cells. Does that help?
Yeah.
So four ways of how a cytotoxic T cell kills abnormal cells. The virus invades the whole cell. Okay, this is the first step. Virus invades the whole cell. A whole cell contains a nucleus, and the viral antigen that the virus hijacks the cell, makes the cell make it in the viral proteins. And the result is that some of those viral proteins are attached to an MHC1 molecule and put on the surface of the cell, because that's just how cell development works. They always put up some proteins. So what they do is they put one of the viral proteins out there. The cytotoxic T cell recognizes and binds with the foreign antigen. And it knows that that is a foreign antigen. It's a viral. It's not one of your self proteins. It's a viral protein. And so the cytotoxic T cell is going to release chemicals that are going to destroy that viral infected cell before the virus can enter the nucleus and start to replicate. Questions? Well, what's its mechanism of action when it comes to killing these cells? The killer cell binds to the target, binds to that MHC1 molecule that has the viral antigen attached to it. And as a result of the binding, the killer cells perforin, remember we saw this with natural killer cells, so we see it also with cytotoxic T cells. Perforin containing granules fuse with the plasma membrane of the cytotoxic T cell. The granules are then disgorge their perforin, and that perforin floats over to the viral infected cell and inserts itself in the viral infected cell's plasma membrane. And the pores emit water and salt, causing the cell to swell and burst and die. So the major difference between this and the way that natural killer cells work is this isn't going to secrete granzymes. It's just going to use the perforins, and the perforins are going to cause the cell to lyse and die. So perforin, of these choices, perforin is released by what?
C.
Are you sure? What's C? Becky, what's C?
Let's see.
What does MHC stand for?
It is major histocompatibility complex.
You think it's going to release perforin?
Well, probably not.
No, it's not going to. What else could release it?
Is it D?
Say it one more time.
D as in dog?
Why do you say that?
Because
Oh yeah.
the natural killer cells, right?
The natural killer cells and cytotoxic T cells do both release this.
Yes.
Okay. So why do we like helper T cells? We love helper T cells. They're awesome. And that when they get activated, they then activate a bunch of other things, and they enhance and activate and stimulate other cells of the immune system. They secrete macrophage activating factors, so more phagocyte activity. They promote phagocytes to move towards sites of infection and inflammation. They secrete interleukin. Remember, that's a hormone that's going to work in between the different leukocytes. So it helps helper T cells. Helper T cells secrete interleukin, which then stimulates B cells. Interleukin can also stimulate cytotoxic T cell clonal selection. There's two mechanisms by which helper T cells can act. The first one is that they bind to an MHC2. They get activated by an antigen presenting cell. And once they're activated, they then are capable of binding to they're capable of activating B cells. With the antigen that's capable of activating those B cells. Remember, the B cells have their little IgD receptors on them. When they're able to bind to a T cell that has that antigen bound to it, it activates these B cells. So they don't have to just randomly run into the antigen. Activated T cells can carry the antigen to the B cell. When this happens, the B cells proliferate and differentiate into memory cells and plasma cells. One thing that's key here is the only way, I can't remember where it is, so I have to go all the way back. The only way that we get memory cells by most of our antigens is if there's a T helper cell. Actually, the only way we get memory cells is through a T helper cell activation. This is important because this is one of the major reasons why we like T helper cells, is because the only way that we're going to create a memory, an immunological memory through creating memory cells, is through activation by helper T cells. The only way that we're going to create memory cytotoxic T cells is through interactions with helper T cells. The only way that a flu vaccine works as well as it does is by allowing you to generate memory cells, so that in a couple months, if you're re-exposed to that antigen of that flu virus, you can combat it again, even though you no longer have plasma cells that are secreting antibodies against that flu virus. Leo, you can't be up there, bud. So this is the first way. They interact with a B cell. B cell proliferates, creates memory cells and plasma cells. So how does this work? How does this interaction occur? So immunocompetent B cells are exposed to an antigen. The antigen binds only to B cells with complementary receptors. The B cell then internalizes that antigen and displays it as a processed epitope. The helper T cell then binds to the B cell and secretes interleukin 2. stay down here. Clonal selection stimulates the B cell to divide and repeatedly form clones. Stay down, don't get up there. Some of those clones become memory cells and some become plasma cells. Those plasma cells then go on to synthesize and secrete antibody, and then can neutralize the antigens. Second mechanism of action by helper T cells. Cytotoxic T cell comes in contact with an infected cell. Actually, wait, let me back up. The helper T cell comes in activation with and to proximity and gets activated by an antigen presenting cell. It stimulates interleukin-2. The same thing that the helper T cell uses to activate B cells. So helper T cell secretes interleukin-2. As interleukin-2 is moving around in the bloodstream and floating around, during that time, a killer T cell comes into contact with an MHC1 molecule that has a viral antigen or a cancerous, probably more, not so much cancerous, but viral antigen. Leo, stay down. And the result is that this killer T cell, when it gets activated and also comes in contact with interleukin-2 that was secreted by the helper T cell, it undergoes clonal selection and proliferation, where some of those also become memory T cells. And the clonal selection then can go out and kill all of the viral infected cells, all the proliferated killer T cells. Questions? Leo, no, buddy. You put a hole in my shirt, I'm gonna be mad. You're so why is AIDS and the HIV virus so nasty? Because it attacks the T helper cells, which, as we've learned, is a major coordinator of innate humoral and cellular immunities. Oh, once you destroy helper T cells, you no longer are able to create immunological memory. Remember, in each case, when you make memory cytotoxic T cells, when you make memory B cells, in both cases, you have to have helper T cells around. When you eliminate the helper T cells, you no longer are able to have any immunological memory. So every time you get exposed to an antigen, vaccines are not going to help you. Anytime you get exposed to an antigen, your only response is the primary response every time. typically people with AIDS die of infections that your immune system should easily have gotten rid of. A lot of times they're herpes simplex viruses that your body keeps in check all the time because you've most likely been exposed to it a long time ago. But now you don't have the ability to have any memory cells. Questions? Memory cells participate in both innate immunity and the immune response. Or what blank participates in both innate immunity and immune responses? Leo, no. No, you're a stinker. Somebody tell me, what do you think?
Can you read the question again? I can't see the whole thing.
Yeah. Blank participates in both innate immunity and the adaptive immune response.
I was going to say D.
Say it one more time.
D for dog.
D is in Delta. That's right. Helper T cells recognize antigens when they are bound to what?
I would say D again.
D. Yep. Let me say something really quick. So if you remember, all of the antibodies start with IG, IgA, IgD, IgM, IgG. IG stands for immunoglobulin. IG. So one very important cell that we don't, this is a T cell that we know the least about, is regulatory T cells. There's also called suppressor T cells. We know the least about them, but they are important because inflammation is great as long as it's needed. But when it's not needed, it's just destructive. So T cells suppress immunity once the infection is gone. So once a pathogen is eliminated, regulatory cells inhibit the immune response by inhibiting multiplication and cytokine secretion by other T cells. So they inhibit the proliferation and secretion by other cells. They downregulate cytotoxic T activity, cytotoxic T cell activity, to prevent excessive production of inflammatory cytokines that could cause extensive damage and possible autoimmunity. So a comparison and contrast side of B cells and T cells. B cells maturate in the bone marrow, mature in the bone marrow. T cells mature in the thymus. Receptors for an antigen on the B lymphocytes are the B cell receptors, or BCRs, or what they're also called, which are antibodies inserted into the plasma membrane that are highly specific. T cell receptors, or TCRs, are present on the plasma membrane, but are not the same as antibodies. You notice there's one antibody receptor or one antibody binding site on the T cell receptor and two on the B cell receptor. B cell lymphocytes bind to extracellular antigens such as bacteria. T cell receptors bind to foreign antigens that are bound to self-antigens such as virus-infected cells. B cells active B cells become plasma cells that secrete antibodies. When T lymphocytes are activated, they become either cytotoxic, helper, or regulatory T cells. They both are capable of forming memory cells. B cells are involved in antibody-mediated immunity. T cells are involved in cell-mediated immunity. B cells secrete antibodies. T lymphocytes or T cells secrete cytokines. B lymphocytes help eliminate foreign invaders by enhancing the innate immune responses against them and provide immunity against most bacteria and a few viruses. T lymphocytes lyse infected cells and cancer cells and provide immunity against most viruses and a few bacteria. And they aid B cells in antibody production and modulate immune responses. Questions? I'm not going to read this. This is kind of excuse me, let me see, a review of what we've been talking about so far. Some more summary slides. A lot of this is just a summary of what we've been discussing. Which of the following, and this is, I know this was a while ago, so I'm going to make you work for it. This was last week. Which of the following does not belong to the second line of defense? Remember, there were 3 lines of defense. Which one does not belong to the second line of defense?
Is it C?
C is in Charlie?
Yeah.
Nope, that's on the first line of defense. What else could it be?
D as in dog.
Why do you say it's D as in dog, Becky?
Just I guess process of elimination. Inflammation is like a sign of, that your body's working to get something out. Natural cells will go after something. The macrophage system will also attack unwanted items. I just feel like D just doesn't belong.
You're right. And think of it, gastric juices are pH.
Right.
And this is on the primary line of defense. Something comes into your stomach that could potentially make you sick.
Right.
Yeah. And the first thing it does is the pH of your stomach kills it. What lacks the capacity to remember a pathogen or react differently to it in the future? Whereas blank utilizes memory cells to adapt to a given pathogen and ward it off more easily in the future.
I would say D.
D is in Delta, is that right, Martin?
Yeah.
Why do you say that?
Because I remember like adaptive was just like it related to B cells, and B cells have memory B cells, which are the ones that basically like remember the antigens from before.
You're awesome because that's right. That's correct. All right, now we're getting into last final details about the immune system here. Tolerance refers, immune system tolerance refers to preventing the immune system from attacking one's own tissues. So a major failure, the failure of immune system tolerance is autoimmunity, where your body starts attacking its own tissues. Mechanisms involved in tolerance is clonal deletion, getting rid of cells that have been activated that could possibly destroy your own tissues. Receptor editing, inhibition by regulatory T cells, and ignoring certain things. Allergies are not an immune system tolerance. Your immune system is going up against something that actually is not harmful to you. And immune privilege, letting certain things just slide. So autoimmune diseases result from the loss of tolerance to self-antigens. Over 100 different types of autoimmune disorders. Leo, no. Some examples of what could cause them: exposure of a normally inaccessible self-antigen that induces an autoimmune attack against these antigens. So something that's normally inaccessible induces an immune attack. Normal self-antigens can be modified by factors such as drugs, environmental chemicals, or viruses, or genetic mutations so that they are no longer recognized as being part of you. And therefore they're destroyed, they activate the immune system. Exposure of the immune system to a foreign antigen that's identical to a self-antigen. Structurally identical. Lingering fetal cells of a fetus within a mother who's no longer pregnant that create autoimmunity because they're somewhat related to you still. Immune surveillance. So within this time period that we've been sitting here, what, almost 2.5 hours, your body has sensed and destroyed a possible tumor cell. So some cell in your body decided it was going to override the checkpoints involved in cell division, and your body has already destroyed it. By one of your cytotoxic T cells saw it and got rid of it for you. This is the process so immunosurveillance is the process by which T cells recognize and destroy newly arisen, potentially cancerous cells. This is a clone of cells. A tumor is a clone of cells that's identical to the original mutated cell. A benign tumor will not infiltrate surrounding tissues. A malignant tumor, in contrast, is invasive will infiltrate the surrounding tissues, and those cells can spread to other tissues that are much further away and may spread throughout the body and cannot be removed surgically. This is an awesome picture. A T cell destroying a cancerous cell. So how do these cells do it? We have macrophages, natural killer cells, and cytotoxic T cells that are capable of killing viral infected cells and cancerous cells. Cytotoxic T cells, natural killer cells. The cytotoxic T cells, natural killer cells, put out toxic chemicals and kill and attack the cells. Macrophages phagocytize it and destroy it. In the process of them killing these cells by either secreting toxic chemicals or by phagocytosis, they secrete interferon, preventing multiplication of other cells that could be cancerous. Interferon also enhances cyto T cell activation, natural killer cells, and macrophages. So immunosurveillance is a process in which non-specifically detect and destroy foreign cells, diseased host cells. What do you say? It would be on this slide right here.
A?
I would say A2. Some immune diseases. You either have two different types. You have immunodeficiency diseases where your immune system is just not strong enough, and there's too little of an immune response, or you have inappropriate immune attacks. Immune deficiency examples are severe combined immunodeficiency, or SIDS, SCID. Not SIDS, it shouldn't be we gotta be careful not to, because SIDS is sudden infant death syndrome. SID is severe combined immunodeficiency, or AIDS, acquired immunodeficiency syndrome. Inappropriate immune attacks, too much or mistargeted immune responses. An example, autoimmune responses or immune complex diseases or allergies. Allergies is an acquisition of an inappropriate specific immune reactivity or hypersensitivity to a normally harmless environmental substance. You're like me and you deal with allergies like around the clock sometimes. I take an allergy med, which helps me 99% of the time and keeps mine down, and they'd probably be much worse if I didn't take it. But I can tell you yesterday, my nose was running the whole day long.
Oh, I have it too. It's
I know
terrible.
I think it's mine. I mean, I don't even really know what it is. It's wind and it's mold. And if there's snow is all over the ground outside, I don't deal with any allergies. And so I'm pretty sure it's a mold. But
Once it starts getting warm, it's terrible.
oh, it's awful. But so hypersensitivity to something that was normally harmless. There's two categories. There's an immediate hypersensitivity and a delayed hypersensitivity. Immediate hypersensitivity, you come in contact with it, it gets active, a B lymphocyte binds to that allergen and it releases IgE antibodies. Think of IgE antibodies as being your allergy antibodies. IgEs then activate mast cells to release histamine granules that then stimulate your allergic reaction. Make your nose run the whole entire day. Make you feel like you have a cold when you don't. Different mediators that can be released from mast cells mast cells are histamine, slow reactive substance of anaphylaxis or SRSA, eosinophil chemotactic factor. Chemotaxis meaning moving towards a chemical. So a chemotactic factor for eosinophils is a factor that's going to be a siren to eosinophils to come that direction. Wherever the siren is, the eosinophils are going to be attracted towards it. Leo, no. Symptoms vary depending on the site and allergen and mediators involved. Most symptoms are hay fever, asthma, or hives. Anaphylaxis, an anaphylactic reaction or local anaphylaxis is an immediate acute hypersensitivity that most often can be relieved by antihistamines. I'm gonna add that most often because one of my partners in crime for anatomy and physiology is allergic to Benadryl. So that will actually initiate an anaphylactic shock in her to even try to give her Benadryl, which is an antihistamine. The antigen triggers the production of those IgE antibodies. The IgE binds to the surface of a mast cell or a basophil. Next time, because now you've got memory cells. The next time it happens, you get an even bigger, stronger reaction than you had before. And the result is that the histamine increases the permeability and you get dilation of blood vessels and swelling. Anaphylactic shock is severe widespread acute hypersensitivity spread by bronchial constriction, dyspnea, which is difficulty breathing, widespread vasodilation, circulatory shock, and sometimes sudden death. Symptoms can be relieved by epinephrine. Stay with me for 4 more minutes here. 5 steps to describe the roles of IgE and mast cells in hypersensitivity. All these antigens enter the body for the first time. The allergens bind to the B cell receptor. the B cells now process the allergens, and with the stimulation by helper T cells, proceed through the steps of clonal expansion of the activated cells. The activated plasma cells then secrete IgE antibodies that are active against the allergens. The FC, remember the FC tail portion, remember that's the constant portion of the antibody, or its tail end. Binds to the IgE tail receptors on mast cells or basophils. Here's your mast cell. And unlike B cells, the mast cell can then have a variety of antibody surface receptors. When an allergen enters the body upon a subsequent exposure, it binds its matching IgE antibodies on the mast cells, stimulating the mast cell to release histamine and other substances. Any allergen that's preformed matching the IgE are attached to the mast cell and can trigger histamine release. So you can get multiple allergen IgE receptors bound to the mast cell. So in one allergen, different allergens can all activate one mast cell. And the first exposure is what allows the IgE antibodies to actually bind the allergen and then become part of mast cells. That's why your first exposure is not that bad, and it's your second exposure that's much worse. Because now you've got mast cells that are ready for it the next time it comes around. I'm just going to try to get through a little bit more of this. We're almost done with the immune system here. So bear with me here for the next. Actually, you know what? Nope, we're done. We will finish this. We might finish it after we get done with the case studies, because we only have 10 slides left. Or we may just work on the case studies, because it might stimulate some interesting conversations, these case studies. But for right now, I will see you at noon.
See you then. Thank you.
Welcome.
See you guys. Thanks.
Thank you.
See you then. Bye.
Thank you.
Welcome.
Thank you.
Hi, Professor.
Hi.
This is Olu.
Hi. I saw your email. So
Oh, okay.
when are you leaving?
The 27th of February. It's for 1 week. Let me check again. Sorry.
Oh, it's coming up. So 2 weeks from now?
Correct.
Okay. Are you
right.
are you gonna have internet access?
I'm not really sure. That's why we're told to send that to our school.
I understand. I understand. There are always recordings posted of lecture and lab.
Okay.
Is there other things that I can do to help you outside of
No,
deadline?
that's fine. I mean, just to let you know, that's the reason why.
No, I appreciate you letting me know. And if there's things that you need me to do to help you when you get back,
Right.
let me know, okay?
Right. Thank you very much.
Of course.
Right.
How do you say your name? Help me out.
It's all me. Just call me Olu. It's
Olu.
Olumuyiwa.
Okay, see, I can do that. All right, I got that. Olu, let me know if you need help when you get back, all right?
Sure. All right. Thank you, man.
of course.
All right, see you.
See you in a little bit.
Okay.
Alexa or Katie or Alyssa, are you here?